Hello everyone. So today I'm going to discuss about the drug metabolism. So what is biotransformation? So chemical alternation of the drug in body that converts non-polar or lipid soluble compounds to polar or lipid insoluble compound, that is known as biotransformations. So what are the consequences of biotransformations? So active drug sometimes becomes to inactive metabolite. Example is pentobarbital, morphine, and then chloramphenicol. Sometimes active drugs becomes active metabolite like phenacetin, and sometimes inactive drugs becomes active metabolites, L-dopa or levodopa. So let us know what is pro-drugs first. So inactive drugs is converted to active metabolite. Okay, so this was first discovered by Albert. Okay, in year of 1958. So what are the advantages? So it is increased. It increased the absorption. Okay, of the drug molecule. Elimination of an unpleasant taste. Okay, sometimes decrease the toxicity of the drug associated with, and then decreased metabolic inactivation, and then increased chemical stability, and then prolonged or shorten the actions. So there are two main phases of drug metabolism. One is a phase one and phase two. So let us know what is phase one and what are the process involved in phase one and phase two drug metabolism. So in phase one drug metabolism, most of the thing functionalization reaction happen here and converts the parent's drug to a more polar metabolite by introducing or unmasking a functional group like hydroxyl, amine, or SH. And in the phase two, the main reaction happens in the phase two is conjugation reactions and subsequent reaction in which a covalent linkage is formed between a functional group on the parent compound or phase one metabolite and an endogenous substrate such as gluconic acid, sulfate, acetate, or an amino acid. So phase on drug metabolism. So what are the reaction involved? We can see. So we can see hydrolytic reaction like ester formation, ester hydrolysis, amide, epoxide, and then arene oxidides by epoxide hydrase. Okay. Then various kind of oxidation reactions comes under the phase one metabolism like aromatic moieties, olefins oxidation, benzylic or allylic carbon atom on C double bond O, C double bond N bond oxidation at aliphatic and acylic carbon, and then C heteroatom system. Okay, and oxidation of alcohols, aldehydes, and miscellaneous. So various oxidation reaction comes under this, and then again reduction. Okay, so aldehydes and ketones reduction, nitro and azo, and then miscellaneous. And then what is the phase two drug metabolism comes like a conjugation reaction, like conjugation like gluconic acid, sulfate, glycine, and ester, other, and then glucothione and mercapturic acid, acetylation, and then methylation. All these reactions come. So, sites of drug metabolism, which part of our body and which metabolism take place. That is good to know. So it's like you can see in so exotherapy like a microorganism enzyme like oxidation and conjugation reaction happen in brain, liver, okay, and all this in lower intestine, and all this we can see. So drug metabolism can occur in all tissues and most biological fluids. Okay, however, the widest use range of metabolic reaction occur in the liver. A more substrate selective range of metabolic process take place in kidney, lungs, brains, placenta, and other tissues. Orally administered drug may be metabolized as soon as they are ingested. However, the first region where a significant degree of drug metabolism occurs is usually the gastrointestinal tract and within the intestinal wall. Once absorbed from the gastrointestinal tract, many potential and existing drugs are extensively metabolized by fast path metabolism. So for example, the fast path metabolism of some drugs like lidocaine is on complete that cannot be administered orally. Bioavailability of other drugs such as nitroglycerol, okay, propanolone, and halide, okay, is significantly reduced by their fast pass metabolism. So phase one or non-synthetic reactions. So oxidation, first reaction as I discussed. So let us discuss in detail. Okay, so addition of oxygen, negatively charged radical, or removal of hydrogen, positively charged radical. Okay, reactions are carried out by group of monooxygenases in the liver. And final step involves cytochrome P45, hemoprotein NADPH, and cytochrome P450 reductase and oxygen. This is the example of phase one oxidation reaction. So you can see there is an enzyme called CYP450 which contains iron 3. Okay, so when a hydrocarbon comes, we can see the overall mechanism and how it is converting the iron to alcohol. So this is oxidizing the RH to ROH. So overall reaction is RH, oxygen, plus H plus NADPH, and it is converting RH to ROH and converting and getting byproduct water. Okay. So oxidation of aliphatic CH bond involving the cytochrome P40, P40 450 family is believed to take place in a series of interrelated steps. So these steps are categorized by cytochrome P450, cytochrome P450 reductase, and cytochrome B5 reductase with either NADPH or NADH as a coenzyme. So cyclochrome P450 is a heme protein molecule in which the heme like the iron reduce the porphyrin IX containing coordinated iron. When the system is at the rest, the iron is fully oxidized to Fe3 plus state. It has been proposed that the substrate initially binds the active site. This is followed by a reduction of Fe3 plus of cytochrome P 450 to Fe2 plus state by one electron transfer catalyzed by P4, P450 reductase. At this point, the molecular oxygen bind to the Fe2 plus and it is converted to the series of other reactions and gives the finally alcohol. So flavin monooxygenase, another examples, are also important family of non-selective mixed functional oxidase. These enzymes which have flavin adenine dinucleotide prosthetic group require either NADH or NADPH as coenzyme. They catalyze the oxidation of nucleophilic groups such as aromatic ring, amine, thiol, and sulfide, but will not metabolize substrate with anionic group like cytochrome P40, P450, and the metabolic ephemerocatalysis. Oxidation is thought to be to involve a number of oxidative and reductive steps. The flavin residue is believed to play a major part in the oxidation, which is thought to occur by attack to the nucleophile on the terminal oxygen and hydroxide residue from on the residue. A number of other enzymes such as monoamine oxidase, alcohol deoxygenase, and lanthanum oxidase drug metabolism. These enzymes tend to be more specific, oxidizing xenobiotics related to the normal enzymes. Reduction. So this also comes under the phase one reaction. So reduction is an important reaction for the metabolism of compound that contain reducible groups such as aldehyde, ketone, alkene, nitro compounds, azo compound, and sulfoxide. The products of many of these reduction are functional group that subsequently could undergo further phase two reactions to form derivative that are more water soluble than the original drug. So reduction of some functional groups results in the formation of stereoisomers. Although this suggests that two metabolic routes may be necessary to deal with the product of the reduction, only one product usually predominates. So, for example, R plus warfarin is reduced to mixture of corresponding RNase and RNA diastereomer, and RNase isomer being major product. The enzyme used to catalyze metabolic reactions are usually specific in their actions. For example, aldehyde and ketones are reduced by soluble aldoketo reductase, and nitro groups by soluble nitro reductase, and azo group by multi-component micromolar reductase. All these enzymes are found mainly in the liver, but also occur in the kidney and other tissues. Many of these enzymes require NADPH as the coenzyme. So this is the structure of warfarin. So after reduction, you can see it can generate a set of stereoisomers. Okay. So hydrolysis, that is the another reaction that comes in the phase one reaction, drug metabolism. So hydrolysis is an important metabolic reaction for drugs. Those structure contain ester and amide groups. All types of ester and amide can be metabolized by this route. Ester hydrolysis is often catalyzed by non-specific esterase in the liver, kidney, and other tissue, as well as pseudo cholesterol. So in plasma, okay. So amide hydrolysis is also catalyzed by non-specific esterase as well as by non-specific amidase, carbopeptidases, aminopeptidases, and declyrases. So these enzymes are found in various tissues in the body. More specific enzyme system are able to hydrolyze sulfate and gluconate conjugate as well as epoxide glucose, etc. So this is one of the example of hydration reaction. You can see that paracetamol is getting hydrolyzed to amino acid, okay, like para-amino benzoic acid, okay, and it happen in liver and intestine. Example, cholinesterol. Other phase one reaction. This is the example. L-dopa is converting to active dopamine by removing the carbon dioxide. Phase two. Phase two is most of the reactions are conjugation reaction because they involve in attachment of a group or a molecule to the drug or metabolite. They may occur at the point of metabolism of a drug or xenobiotic, but they are often the final step in the metabolic pathway before excretion. So the product formed by this reaction are known as conjugates. They are normally water soluble and are usually excreted in the urine or the bile. Okay, so the conjugates form are usually pharmacologically inactive, although there are some notable exceptions. Okay, for example, it has been suggested that the hepatotoxicity and neuronephrotoxicity of phenolphthalein could be due to the formation of O-sulfate esterase, which has been shown to bind to the microsome protein. So this is the reaction. Okay, these are the other common examples of phase two reaction, like in uh, by different enzymes like L-acetyl transferase enzyme. It is converting amine to amide, and then here sulfonate transferase converting amine to a sulfonate salt. Okay, this is more examples. Okay, where a carboxylic acid group is getting converted to a ester. Okay, and again here a amide bond formation happening with a amide group and a acid group. So biological factor affecting the metabolism. So the various factors affect the drug metabolism. So one of them is age. The ability to metabolize a drug is lower in very young age. Okay, under 5 and elderly like over 60. However, it is emphasized that the quoted age are approximate. Okay, and our actual changes may vary according to the individual and their lifestyle. If the fetus and the very young nominate very metabolic routes are not fully developed. We all know this is because the enzymes required to metabolic processes are not produced in sufficient quantity until several months. Okay. So, for examples, when chloramphenicol was used to treat a bacterial infection of premature babies, it was found to have a high mortality rate, which fell considerably when the babies were 30 days old, because the enzyme required it is not developed, and that's why it was not able to metabolize the chloramphenicol. So some xenobiotics are able to cross the placenta from the mother to the fetus, and so any drug used in the mother are also like to pass through into the fetus. Okay. So since a number of enzyme systems that are required by the body for phase two drug metabolism reaction have been found to be present in a negligible or low concentration in the fetus, this drug can affect biological processes in developing fetus, resulting in teratogenic and other undesirable effects. Furthermore, where drugs metabolism occur, the more water-soluble metabolites of the drug are likely to accumulate on the fetal side of the placenta. Obviously, if these metabolic products are pharmacologically active, they could also be detrimental to the development of the fetus. Children above 5 and teenagers usually have some metabolic route as adults. However, this smaller body volume means that smaller doses are required to achieve the desired therapeutic effect. It is well documented that a person's ability to metabolize drug decreases with age. This decline in the person's ability to metabolize drug has been attributed to the physiological changes that accompany aging. Okay, it does not pose to many problems because the approximate ages between 20 to 60 years, but can become more significant in the elderly. In general, the body gradually loses its capacity to metabolize and eliminate the drug and its metabolites. So this leads to higher blood concentration in the drug and possibly of an increase in the adverse effect of the drug. This is normally encountered by giving reduced doses of relevant drug to the patients. So you can see that this is a variation of the blood metabolite in the drug. Okay, examples. Gender. So the metabolic pathway followed by drug is normally the same as both males and females. However, some gender-related differences in the metabolism of anxiolytics, chronesics, and a number of other drugs have been observed. For example, Wilson has found that diazepam has an average half lifetime of 41.9 hours in female, but only 32.5 hours in males. Such differences have been attributed in some instances to significant differences in enzyme concentration. For example, women have a significantly lower concentration of alcohol dehydrogenase and so do not metabolize alcohol so rapidly as men. The pregnant woman will also exhibit changes in the rate of metabolism of some drugs. For example, the metabolism of analgesic pethidine and antipsychotic comaprazole is reduced during the pregnancy. So these are the structure of this drug molecule. Genetic variations. So variation of genetic codes of individual can result in the absence of enzymes, low concentration of enzymes, or the formation of enzymes with reduced activity. This difference in enzyme concentration and activity results in individual exhibiting different metabolic rate, and in some cases different pharmacological responses for the same drug molecule. For example, the anti-tuberculosis drug isoniazid is metabolized by phase 2 reaction, acetylation. In some patients, this acetylation is fast, in others, it is slow. Slow acetylation is found in 75% Caucasians and Negroes, but in only 10% in Japanese and Eskimos. Patients are often classified as fast and slow acylated. Okay, so environmental factor affecting metabolism. The metabolism of drug was also affected by lifestyle, poor diet, drinking, smoking, and drug abuse may also have an influence on the rate of drug metabolism. The use of over-the-counter self-medicant may also affect the rate of the metabolism of endogenous ligand or a prescribed drug. Since the ligand use of over-the-counter medication is widespread, it can be difficult to assess the results of some large-scale clinical trials. These are specific examples. So, drug metabolism and drug design. So, a knowledge of the drug metabolic pathway of a drug may be used to design analog with the different metabolism to that of the lead. Okay, so this changes in metabolism is achieved by modifying structure of the lead. These structural changes may neither make the analog more stable or increase, or it is of metabolism relative to the lead. The structure modifications should be selected so that they do not change the natural and pharmacological activity of the drug. And finally, the ease of metabolism of drug may be increased by incorporating metabolically labile groups such as ester in the structure of the drug. This type of approach is the basic of product design. It is also lead to development of so-called soft drugs. So changing the metabolic pathway of a drug may also be used to develop analog that do not have the undesirable side effect of the lead compound. For example, the local anesthetic lignocaine is used as an antiemetic. In this respect, it is undesirable covasculant and ultimate side effects are caused by its metabolism in the liver by dealkylation to the mono-N-ethyl derivative. The removal of N-ethyl substitution and their replacement by alpha-methyl ester gives the anti-aromatic toconamide. The toconamide cannot be metabolized by the same pathway as the ligocaine and does not exhibit covasculant and intimate side effects. So this is the overall reaction happens. Thank you. Thank you for listening.